Welcome to the JGI Genomes of Energy, Relevant for Energy and Environment, Genomics for Energy and Environment. This is, I think, the 9th of these meetings we've been having. This is the biggest crowd we had, which is great. This meeting has a variety of, and I'm Eddie Rubin, I'm the director of the JGI. This meeting has a variety of purposes. Obviously a central purpose is to hear a lot of neat science using genomics loosely connected to energy and the environment. Very loosely, you'll hear from some of the talks. But another purpose of the meeting is the JGI is first and foremost a user facility. And we are very different now than we were 5 years ago, and we're going to be very different 5 years from now. And if we're not going to exist. And so we need to continually evolve because there's just changing technologies, and what genomics offers to us is changing. And the directions we go is in large part determined by our users. And so this is an opportunity to let us know what kind of capabilities you would like to see the JGI develop in the future. What kind of user programs you'd like to see the JGI in the future. And so you see a lot of people that will have JGI on their name tags, and I urge you to use the next 2.5 days to let them know what you're thinking. And then there is a session Thursday afternoon where we'll have an open mic where will be another opportunity to tell us where directions you'd like to see the JGI go. And so it's a pleasure to have Steve Quake here. Steve, this is sort of payback. He's here 20 minutes late because he spoke about 6 years ago at this meeting, and it was the last session, and there was about 20 people in the audience. And so now we got a full room, and so we wanted to make sure everyone was here before he started. And so it is a pleasure to have Steve here. Steve is a professor at Stanford. He's really took a path from, his academic path was from Stanford to Oxford to Stanford. He did a postdoc with Steve Chu, many of the people here know, and then eventually took his first faculty appointment at Caltech, and then moved back to Stanford to help head up the bioengineering department there. And he's had really a terrific productive career. He's received many of the big prizes. He's a member of been elected to all the academies and societies. But I think one thing quite unique about Steve has been his ability to move the science and technology in his lab to have it commercialized, so eventually it's at the end of our lab bench. And Steve has been the founder or co-founder of more than a half dozen companies, and many of these companies became really big. In fact, one of them is Fluidigm. When Steve was at Caltech, he was trying to do for moving fluids a little bit like the electronics industry did, and developed microfluidic chips that have really enabled lots of science. And so that was a success story. One of my favorite companies he started was Helicose, which is a DNA sequencing company. They made a sequencer. It wasn't a success story. But it was really cool technology. It was a single molecule sequencer. It had really a lot of great features, and there was this evolutionary bottleneck in sequencing, and it just did not find its way through that bottleneck. But if they had maybe had different operations and management, instead of being, Illumina being the gorilla that came out of the bottleneck, it might have been Helicose. And so the company that Steve founded. And so it's a great pleasure to have Steve here talking. Steve is working with the JGI, and one of the ways the JGI is evolving is that we're progressively offering new capabilities and new technologies to users. And many of those we're developing in-house at the JGI, but also we're working with very creative technologists that are developing things outside the JGI with the hope that we'll bring it inside and make it available to our users. And so Steve will be talking about single cells.
Well, thanks so much for that very generous introduction. I do apologize for being a bit late. I'm still kind of dazed and confused by all the traffic. I knew this was a really big meeting. I had no idea that people would be coming from miles around, clogging up the freeways trying to get in here. So I'm going to talk today about single-cell genomics, which is sort of a field that's having its moment right now. It's getting a lot of attention, and we've been sort of digging away at it for a bunch of years. And it's interesting that it's actually a very old field, and it's more than 100 years old. It sort of got its start back in the late 19th century when Walter Fleming noticed that there are these enormous molecules inside the glands of some mites and insects, and people from the Drosophila world will be very familiar with this phenomenon. It's called polytene chromosomes, happens in the salivary glands of Drosophila, and it was something that was exploited to very practical ends, starting with the work of Calvin Bridges at Caltech back in the 30s, who actually mapped the genome using these very highly polyploid condensed chromosomes from salivary glands, where you could sort of take pictures from single cells and work it out. And this was used for decades to map out differences between fly lines and species and so forth. And so, what's all the excitement now? Well, there's really been, I think, 3 key advances that have all come together to dramatically increase the power of these sorts of approaches. One has been the development of biochemical amplifiers, such as PCR and MDA, date back to the 80s. And almost as soon as PCR was invented, people began to think, gosh, we ought to use this to look at single cells and dissect what's going on. There's a literature that sort of dates to that period, and it's sort of very interesting literature. And it again didn't really take hold the way it could have because the other things didn't happen. One of which was the development of high-throughput sequencing technologies, which have dramatically lowered the cost of how to sequence genomes, analyze transcriptomes, and basically read out nucleic acids with sort of arbitrary depth and complexity. And the third piece, which I think has also been crucially important, has been the development of single-cell manipulation technologies. And FACS, fluorescent activated cell sorting, is sort of one of the first ones. Microfluidics has been another, and I think there's going to be sort of a continuation of advances in these sorts of technologies over the next few years, and you'll see a lot of cleverness and creativity there. And you combine all 3 of these things together, and all of a sudden, you can do a lot. And this was sort of recognized last year by Nature Methods, who declared it the method of the year, which they announced in their succeeding year January issue. So if you look at the January 2014 issue of Nature Methods, there's a number of nice commentary articles that sort of give different perspectives about where people think the field is going. So as Eddie mentioned, I got into this whole field. I used to do single molecule biophysics, and we developed an assay for DNA polymerase that led to the first single molecule sequencing, and a sequencer that, when it was launched, it was the world's fastest, cheapest sequencer. And we did get outmaneuvered by Illumina, sadly, but not before we did a lot of good work with the machine. And it took Illumina years to catch up, actually, with the parallelism and throughput that we had done back then. And that sort of pulled me into this whole field of genomics, and I realized that we could sort of very productively combine these sequencing technologies with these sort of tools for automated plumbing that we had also been working on in parallel, which we called microfluidics. And this is something we think about as the integrated circuit of biology. And it's a way to sort of automate fluidic steps. So we have chips that instead of having wires and transistors, they've got pipes and valves and pumps and so forth, and we can create any sort of plumber's nightmare you like from that. And one of the devices that became very useful for single cell analysis was one that was developed by my then student, Jian Liu, who used the plumbing to create a combinatoric array, a way to do lots of experiments at once. And this array was a matrix, and there's a bunch of rows and columns, and everywhere there was a vertex where a row and column intersected, you could do an experiment. And so this allowed you to do, for example, PCR, where you put in lots of different templates, lots of different primers, and do all possible combinations of them. His initial chip was 20 by 20, so it could do 400 experiments with 40 pipetting steps. And this eventually got commercialized by Fluidigm, who built versions that did 48 by 48 and then 96 by 96. These are sort of the world's highest throughput PCR machines, and they became a great tool for analyzing single cell gene expression, where we'd put lysates from the single cells in all the rows and primer sets in all the columns, so you could look at 96 genes per cell across 96 different single cells and get quite a bit of data from each chip. And so we sort of embarked on a program of trying to analyze single cell gene expression. Excuse me, I'm not going to totally focus this on microbiology, microbial genomics. Eddie sort of gave us license to talk about whatever we were interested in, but pardon my sort of digression. But you'll see it'll loop back, I think, to the things of interest here. So we began trying to ask questions about noise. And we were interested both in the technical noise of the measurements, sort of how well could we measure gene expression from a single cell, but also what were the sort of sources and role of biological noise in gene expression. And this is something that's been really intensely investigated in the last decade, primarily in bacteria, but folks like Mike Elowitz, Ido Golding, Sunny Shi, and others. A bit in eukaryotes, people like Jim Hasty, Jim Collins, Alex van Leeuwenhoek, but not quite as much has been known in eukaryotes. And there's this curious result by Sunny and his collaborators that when they looked in bacteria and tried to do single-cell measurements of RNA and protein of the same genes, they found they were completely uncorrelated. That was a bit disturbing. And so we had sort of that in the back of our mind because it really called into question one's ability to use RNA as a proxy for gene product or protein levels. Another thing that was in our mind was the fact that everybody who had looked at gene expression, even the literature that dated back to the 80s and the early days of PCR, people found very broad distributions of transcripts for a given gene, even for boring genes like housekeeping genes. And so these were so broad they appeared to be log normal. And what was that all about? Why isn't the cell regulating this better? And then what are correlations between different genes? And so we began to try to tease this apart in a model system. And we focused initially on a human cell line, HCT116, and decided to try to really tear it apart as best we could. There's been, because this has been an area of some activity, there are some nice models out there to set one's expectation. And sort of the simplest realistic model that had been developed is the one that's shown here, where DNA makes RNA makes protein at different rates. The products get degraded at different rates. And the DNA can switch between active and inactive states, which you can imagine as chromatin opening and closing, transcription factors binding, falling off, something like that. And that happens with some rate. And so you can take all those rates and those arrows in the picture there and write down equations, write down a master equation for this model. You can compute moments of the variable, and you can sort of make approximate calculations of some of the distributions. And this model predicts that in some approximate limit, the RNA distribution would be described by this gamma function here. And we decided to go out and look at that. And this gamma function is a very broad distribution. And in other limits, it does sort of itself become approximated by log normal distribution. So that was interesting. The technical details of the measurements, I'm not going to dwell on here. I'm happy to discuss them later. We sort of titrated RNA standards from bulk RNA down to single cell levels, measured these titration curves. We're able to show that the sort of technical replicates of these had a much lower standard deviation than the actual distribution of expression values in cells. And this is sort of shown here for half a dozen different genes, where one can see here sort of the bulk titrations, and this is the distribution of values from individual HCT116 cells, and the standard deviation shown here of the cells is greater than what you see from the bulk RNA measurements at the same level. And so the technical noise of the measurement is less than the biological noise between the cells, which is a good place to be. And so we went and looked at a lot of different genes, and we fit them to both log normal and gamma distributions, and found that in many cases log normal worked. In a few cases the distributions had some skew to them, and gamma was a better distribution. The p-values are shown here in the top graph right here, where the triangles are the gamma distribution and the squares the log normal. And if it's sort of below the line, that's not a good thing. It says you're fitting to the wrong function, something's wrong. And you can see that gamma's a little bit better, but log normal is pretty good. And so that's a nice picture to have in your head, that the distribution of transcripts should be log normal based on these very sort of simple assumptions about fluctuations in how gene products are made. And there's sort of a rationale for it as well. You don't need the complicated model to think about it. You just return to the central limit theorem, right? Which says if you have a bunch of random variables and you add them up, no matter what their distributions are, you get a Gaussian distribution for the sum. Whereas if you multiply random variables together, what you get is a log normal distribution for the same sort of reasoning, right? You take product of random variables, take the log of the product, that becomes the sum of the logs, which you apply the central limit theorem is normal, so the log is normally distributed, therefore the variable, the product is log normally distributed. And when you think about what's going on in these cells, in that model we wrote down, you're multiplying a bunch of those rate constants together, all of which are fluctuating randomly, and so it sort of all hangs together, and there's a nice intuitive explanation for this log normal-like behavior. And it would be very disappointing if this was all there was to the story, because all we've talked about are random fluctuations here, and nothing about sort of deterministic control of gene expression pathways being turned on and off. And so I'd be very sad if this was, all the data kind of agreed with the model. Unfortunately, some of the data is completely inconsistent with the model, and the RNA levels have much broader distributions. They appear to be even bimodal. And as we sort of dug into that, we discovered some interesting correlations between genes. If we took one of these genes, top 2A, right here, and we divided it up into cells that express at a high level, colored red, and cells at a low level, colored black, and then we looked at other genes and we kept that color coding, you can see that other genes also are expressing, the same cells that are expressing top 2A at a high level are also expressing ANLN at a high level, and other genes as well, and some that are uncorrelated. And that was pretty interesting. And I'll just pause here to point out this is the work of Tomer Kaliski, who was a postdoc in the lab at the time, and now a faculty member at Bar-Ilan University. And so you can compute the correlations across all the genes we measured. So here are the rows and the columns of the genes, and we've correlated across several hundred cells. And one can see that there's some interesting off-diagonal elements of this matrix. And I'll just focus on this one, which is right here. These are sort of the most prominent ones. And these are all genes involved in the cell cycle. And so what we've been able to discover from this is you can see sort of coordinated regulation of the genes involved in the cell cycle for those cells that are dividing. And that also sort of violates the model. And so it's good. We're sort of able to understand something about coordinated expression. How about this issue of correlations between RNA and protein? Well, going back to the model, remember you can compute different moments. And one moment you can compute is the correlation of RNA and protein. And the prediction is shown up here. And you can see that that correlation takes on a wide range of values from 0 to 1. And one of the really important parameters is the mRNA lifetime, measured in units of the protein lifetime. And when that is short, the correlation is 0, and when that is long, the correlation is 1. And so this also is sort of consistent. It's known in bacterial mRNA lifetimes are short, eukaryotic ones are longer, and so one might expect to find different results in these different systems. And sure enough, in our data, we were able to find a reasonably strong correlation between RNA and protein for a couple of things that we looked at. We cloned GFP into these cells, and you could see a very high correlation there. We looked at a couple surface markers. This one is CD49, which is shown here. And you can see that the correlations are non-trivial as we make the scatter plots between all the cells, but also we can correlate the proteins to every transcript we measured, and happily the one with the highest correlation are the ones with the gene product associated with it. Okay, so that's a relief, and we can sort of feel reasonably confident using RNA as a proxy in these single cell measurements. And then we went out and embarked on a sort of program of what I call reverse tissue engineering, trying to use these single cell measurements to work out the hierarchy of complex tissues. And the most famous example of this is the blood, where this whole hierarchy of hematopoietic stem cells down to the various differentiated compartments of the blood has been worked out over a couple of decades. And we thought that this sort of single-cell genomics approach would really accelerate that in other tissues. And so if the sort of conventional approaches have been flow cytometry and immunohistochemistry, we thought we had a really good complement here. And so with collaborations with a few colleagues at Stanford, we've gone and done this in different tissues. The first one I'll tell you about was done in colon with Mike Clark. And the basic experiment is to take crypts, in this case, I'm showing mostly mouse data here, although we've also done some human data, dissociate the cells, label them with antibodies, do FACS, do single cell sorts from the FACS, and then read out the measurements in the dynamic arrays, the PCR arrays that I was describing, and then create these sort of heat maps of clustered dendrograms where we're looking for shared gene expression patterns between different cells. And one of the nice things about working in the colon with the crypts is you can find antibody to surface markers that lets you work out where the cells came from the top or the bottom of the crypt. And there's a very nice connection between geometry and developmental state, where the stem cells all sit at the bottom of the crypt, and as they divide and migrate, as the cells divide and move up to the top, they become progressively more differentiated by the top, they get to the top, they've chosen a fate. They're either goblets or enterocytes, and they get sloughed off, and the whole thing just keeps on cycling. And these crypts turn over about every week in your body. I mean, it's amazing how much cell division is going on there. And so we have this independent measurement for these cells, which we record by fax, of where the cells came from, bottom or top, which corresponds to whether their stem cells are differentiated. And we can use that to sort of calibrate or test these the gene expression profiles. And so what's shown here, every row is a different cell, each column is a different gene, and they've all been clustered together. And you can see that there's different patterns corresponding to different cell types. And over here on the far end are the surface markers that were measured by fax. These weren't used in the clustering at all, and you can see that the gene expression patterns completely separate cells that came from the top from the bottom. So that's good to see. But you can actually see a lot more than that. And we begin to assign names to these different cell types. And we could do that because of the particular genes that were expressed by the cells. So, for example, we called this group right here enterocytes. Oops, let's see. These enterocytes, because they're the ones that are expressing SLC26A3, the gene that's involved in absorption in the colon. And likewise, we call this other group right beneath them to be goblets, because these are the only ones that are expressing mucin. Right? This is the gene that defines a goblet in its differentiated state. And there's sort of two subgroups within the goblets. We discovered an immature group, and I won't dwell on this, but we actually were able to go back and stain for NAV and show that, in fact, there is this immature goblet that's expressing NAV, and it was a nice example of using the gene expression to discover new markers to find a new subset of cells in a hierarchy. We also just mentioned something about the stem cells at the bottom of the crypt. There had been these huge debates in the literature about what were the properties of the stem cells of the crypts. And there was the Hans Cleaver school, which said, oh, the key gene is LGR5. And there's the Mary Kopecky school, which said, oh, the key gene is BMI1. And it turns out they're both right. So stem cells, their population is expressing LGR5 and a different population expressing BMI1. And they're both there at the bottom of the crypt. And really sort of a neat result. And the LGR5 group is expressing genes related to cell division, so they seem to be actively cycling, whereas the BMI1 group is quiescent. And you can see how this sort of just goes on and on and on, and you can have an endless amount of fun, and you can put whatever genes you're interested in the profile, in the chip, and ask all kinds of questions about what's going on with these cells, both in the normal tissue and in diseased tissues, like those tumors derived from cancer. And Mike and I have been sort of pursuing this for several years, both at Stanford and licensed our results to a small company called QuantaCell, which has been trying to use this approach to discover new targets of cancer stem cells to develop small molecule therapeutics against. So we'll see whether that all bears fruit in a few more years. And as we've been doing that, there had also been sort of a small literature percolating along about trying to do whole transcriptome amplification. A few papers were coming out, and so instead of just looking at a defined set of genes, trying to do the whole thing at once. In our hands, we'd been fooling around with it too, and we'd never bothered to publish it because the results were pretty noisy. But other people decided just to do that, and we kind of realized, oh yeah, maybe we should have paid more attention, and the noise wasn't as limited as we thought. So we decided to jump into that game as well. And Fluidigm had just released a chip that sort of automated the whole cell isolation and transcriptome amplification called the C1. And so we decided we would try to compare many of the methods that were out there, both in tubes and in the chip, and see what were their properties. And this is the work of Angela Wu in the lab, was shown here. And we published this in Nature Methods late last year. So this graph just shows sort of in a very gross way what the reproducibility is by asking what fraction of genes are reproduced in multiple experiments and replicates. The first two bars are from bulk experiments with two different reagent sets for making the cDNA and doing the amplification, and the next three are from single cells. And so this one here is from the C1. These two are from two tube-based protocols. And the one on the end here is actually the same chemistry that's done in the C1, but done in microliter volumes. And you can see that they're fairly similar. You're starting to sacrifice something from the bulk, but all of these by this metric are quite similar. But what happens when you compare it against the genes that we measured by PCR? So a subset where we really feel like we understand the measurement, because we worked very hard to understand the technical limitations of the PCR and the sort of the sensitivity and all that. So for that sort of subset of, in this case, I think it was 48 genes. Here's the reproducibility across single cells of PCR. This box and this box. So one is done in the C1, one is done in the microliter volumes with the facts as I described, and they're quite similar. And this is the reproducibility of RNA-seq as done in the C1. And then the others here are reproducibility of the microliter volume amplifications. And you can see that it drops dramatically. So there really is an improvement when you're using a sort of calibrated set of genes in the small volumes. And this is consistent with what we've seen pretty much every time we put a biochemical amplifier into these small volumes, that they tend to work better. For pretty simple physical chemistry reasons. And so that was sort of interesting. We also put in ERCC spike-in controls in sort of every chamber in the chip, and so we can estimate the absolute quantitation. So these RNA controls are a NIST traceable standard. They give you a tube with a defined number of RNA molecules in it of different species. And so we know exactly how many we put into each reaction, and then we can ask how many do we measure. And that's shown here. And you can see that over a very large dynamic range, this is a log scale, you get pretty good absolute quantitation on average. I just want to point out that if you think about what FPKM or RPKM really means, it's sort of this obscure unit that gets used in RNA-seq. And if you want to have an intuitive sense of what it means, when you're at the level of a single cell, 1 FPKM corresponds to more or less 1 transcript or 1 molecule. Okay? You can do the math and work all that out. That's a pretty good number to carry around in your head. And you can see that's more or less what happens here. So 0 and 0, those are 1. 1 molecule in the chip on average of 1 FPKM measured. And you can see that we're having sensitivity down below 1 molecule per chamber. Well, how do we get that? The answer is there's 96 chambers on the chip, and this is an average across all 96. So most of those are empty, and a few of them will have a molecule in them. And when you average all that out, that definitely it does increase the dynamic range and it lets you dig a little deeper. And finally, we begin to compare the absolute levels that we measured by PCR with those we measured by RNA-seq, and found a pretty reasonable correlation. Surprisingly, you try to express both axes in absolute units, the slope ends up to be sort of close to 1. And that gave us some confidence in the absolute values of FPKM we're measuring from these single cells as well, and that we've measured the same thing by 2 different methods is a good way to establish precision and accuracy. That being said, when you start to look at the dispersion, now these are average values across many cells. When you look at the noise, the dispersion, the RNA-seq doesn't do as well. And you're not in that happy situation where the technical noise is less than the biological variation in most cases. In a few cases you are, but generally with the RNA-seq you're dealing with a higher level of technical noise. And so you have to apply it to problems where you're seeing really significant changes in gene expression, I think, to say something sensible, or else have the ability to average across many single cells. And we began to do that and continue this reverse tissue engineering paradigm through a collaboration with Mark Krasnow and Tushar Desai. And this was work done in my lab by my postdoc Barbara Treutlein, and in Mark's lab by Doug Brownfield. And we got interested in trying to sort of ask very similar questions to what we did for the colon, but in the developing lung, in particular during the sacculation process. So when the alveoli are forming and turning into sacs where they can do gas exchange. And there's sort of had been a little bit known, a handful of markers that defined a handful of cell types, and their names are shown here. There's 2 alveolar cells, given the very creative names of alveolar type 1 and alveolar type 2. It indicates something about the lack of knowledge. These gave them numbers. The type 1s are the ones that spread out to be very flat and have this huge surface area, and that's where the gas exchange happens. And the type 2s are these little cubes that end up feeding the other ones, and they're involved in sort of communicating something in a paracrine way. And then there's also in the terminal bronchioli, Clara cells and ciliated cells. They each have a marker associated with them. And so we got interested in trying to figure out if this was all really the case, and if so, what were sort of other genes that were involved, and what were the processes that led to these differentiated cell types. And there was this idea that there must be this bipotent progenitor that led to those 2 alveolar type cells, but nothing was known about it. And so as the sacs form, before they turn into sacs, that progenitor must be there, and then these progenitor cells must choose fates where they either become the type 1s or the type 2s. And we were interested in trying to see if that was really true, and if so, what were the real details of that process. And so the experiment here was to take lungs, either from embryonic days 14, 16, or 18, or from adult mice, isolate the lung, dissect it, digest it, make a single cell suspension, purify away any cells of the immune system that happen to come through, and then analyze the cells in the C1, where we did both whole transcriptome amplification sequencing and qPCR. And it was very nice to be able to go back with an independent method and validate the transcriptome results. And using both together was really quite helpful for us. And so if you start doing unbiased analyses of this data, for example, at E18, if you do principal component analysis, you start to see that the principal components will separate the cells into different groups, which correspond. The markers that were known correspond to different groups here. And there's a third group, which I'll get to later. A fifth group, the bipotent progenitor, I'll come to later. But they start to separate according to the known markers. And if you ask about the loading of the PCA coefficients, you can see that the loading, which is shown here, is definitely the canonical markers are all pointing in different directions. And there's a bunch of other genes that are pointing in those directions that sort of do equally well at differentiating the cell types. And those are all new genes that are uniquely expressed in the different cell populations. And so as we start to create these clustering heat maps based on the genes that contribute most to the loading, you can see that we get the different populations coming out, and you can see the different patterns. Again, the rows are cells, the columns are genes, and each of the canonical markers ends up going in a different group, but along with those canonical markers are many other genes, almost 2 orders of magnitude more than, an order of magnitude more than we started with. So we've got a whole set of new genes that are involved in these different cell types, and it's going to keep several generations of Mark and Tushar's graduate students busy with trying to sort all that out. And I'll tell you a bit about this bipotent progenitor. As we go from day 16 to day 18, there's a huge difference in the PCA loading. This is now from day 18. If you go a day earlier, you can see that the 2 groups, AT1, AT2, are now right on top of each other. So really, this group of cells that were clearly differentiated by the PCA at day 18 can't be differentiated anymore, and the genes that previously differentiated them are now all pointing in the same direction just 2 days earlier. And those are the genes involved, and those are the cells that are the bipotent progenitors. And we went and did quite a bit of more informatic dissection of them, which I'm not going to dwell on right now. But if you're interested in this will all be published soon in Nature. It'll be coming out hopefully in the next few weeks. I want to take the rest of the time to talk about the sort of other half of single-cell genomics, which is not doing the phenotyping or gene expression, but by doing genome analysis. And this was actually something we got involved in quite some years ago, and we realized it would be very useful to use the plumbing to isolate individual microbes, which couldn't be cultured, and try and get genetic information through single-cell genome amplification. And in fact, our first work in this area at the sort of whole genome level was done in collaboration both with Dave Relman and folks at the JGI, where we went after a bug from this candidate phylum TM7 for which there had been previously no genetic information, and we did a very rough draft of the genome and were able to get data on thousands of genes from this phylum. And then we've gone on with a variety of other collaborators, some of whom are here at the meeting today, to do single-cell genomes from things that are interesting from the evolutionary point of view or from their role in the environment and ecology. And that's all sort of steaming along right now. And since a lot of this is published, I'm not going to dwell on it. And I think you'll hear from some of our collaborators over the next few days, so I'm not going to dwell on that. What I am going to do is take a moment for the experts in the audience now. If you're not an expert, you can tune out for a little bit. But for those of you who are experts and really care about sort of the gearhead details of the amplifiers, I just want to tell you a little bit about what we've been doing to understand their properties. And then I'll finish up by talking about how we've been taking this single-cell genome analysis to humans. We've been on all the work we've done, we've been using MDA, which is this amplifier developed by Roger Laskin. It's a beautiful thing. It works isothermally, creates enormous amount of gain, doesn't require any prior knowledge of what you're amplifying, so you have random primers, and it's been sort of great in our hands, and we've been very happy with it. But like in all areas, there's now sort of a lot of creativity in this DNA amplifier business, and there's several other amplifiers floating around now, and there's some that are PCR based. And they seem to have very interesting properties. And the folks who make these amplifiers are now doing a lot of chest beating and saying mine's better than this one. And the waters are getting muddied. So we thought it would be useful to start to do some side-by-side comparisons of the different amplifiers. And so in particular, we decided to compete MDA against two PCR-based amplification schemes. One is called Malbec, came out of Sunny Shee's group, and the other is sold through NEB. It's, I'm blanking on the name of the company that makes it now. Someone help me. But there's a company that makes it. It's a PCR-based amplifier. It's very similar to Malbec. They're quite similar in the details, although there's fine claims about which one is better. So we decided to look at both of them, and we decided to do measurements, amplifications, both on bulk samples and on single cells, and we focused on E. coli. Bacterial genomes are great because they're very inexpensive to sequence, and we could really ask how do these things work on single cells, and what are their various properties. And so one question you can ask is, something about the specificity. How many of the reads you get when you do an amplification are going to map back to the genome you think you started with? And these graphs, green is good. And so, in the bulk samples, you're getting the vast majority of stuff mapping back to the genome you started with. But as you get to the single cell experiments, you can see that green bit falls down precipitously. And let me just this one right here, where about half the reads are mapping specifically, is the MDA result, and then the other two are the PCR-based ones, where you can see that there's just, the other three, let me say that again. This one is the microfluidic MDA, so done in the nanoliter volumes. The next one is the microliter MDA, and then microliter versions of Malbec and the NEB kit. And you can see that as you go to these large volumes, no matter what the amplifier, the performance sort of falls precipitously because it's just impossible to keep the contamination out. And as you start to analyze the contaminants, you can see this is now the reverse graph. This is the amount of contamination. You can see there's a lot of human contamination as well as some other microbes that slip into the reagents. And this is one of the reasons why it's really good to shrink the volume, because you get that under control. Another thing we began to look at was how well you did in covering the genome as a function of the gain of the amplification. So we asked, as you change the gain from 1,000 to 1,000,000 to 1 billion-fold increase in DNA, how does that affect the coverage of the genome? And we did this with a combination of single cell and bulk results. And you can see that this is actually very interesting. The amplifiers behave really differently. And so this one is good, so you want to be up at the top, and you can see that as you increase the gain, MDA is performing worse and worse. I mean, the coverage goes down dramatically, and you see a lot more bias. However, the PCR-based amplifiers are more or less constant as a function of gain. You get the same coverage no matter what, and there's a point where the curves cross each other. Whoops, sorry, let me go back. And there's a point where they cross each other, and that point is more or less by coincidence where we happen to work with the microfluidic devices. And so using the small volumes is a way to limit gain of amplification, and it turns out that whether you do MDA in a microfluidic device or a PCR-based thing in a microliter volume, the uniformity of amplification is pretty comparable there. So that was something that we thought was really sort of striking and a really notable difference in the behavior of the amplifiers. Not that one is better or worse, but you're able to make a sort of informed choice of which one to use based on your experimental design and the questions you're asking there. There has been some results in the literature comparing sort of PCR-based amplifiers and MDA on bulk samples, and one of the results that's been claimed is that for measuring copy number variation, the PCR amplifiers seem to be better. For what they don't cover as much, but for what they cover, you have a better sensitivity for copy number variance. And so we looked at that, and we found the same result, that definitely the PCR-based ones have a more sensitive ability to detect copy number variance. And you can see that here, again, plotted up as a function of gain of amplifier, but the y-axis here is sort of the minimal size of a CNV, the smallest CNV you can detect. And red is MDA, and the other one's Malbec. So you can see for broad ranges, the PCR-based ones are better. However, you can still detect them with MDA, and so people are going around saying you can't do any CNV detection with MDA. That's inaccurate. The final thing we looked at, or not the final thing, the next thing we looked at was looking at the error rate. And so we looked at the error rates as a function of gain for the different amplifiers. And here the MDA is just, sort of quite a bit better, almost an order of magnitude better. So the red data points are MDA. These are the PCR-based ones, which are fairly similar. And you can see that if you care about looking for mutations and single nucleotide polymorphism and so forth, you'll do much, much better with MDA. And this is purely a function of the polymerases that are used, by the way. These numbers are consistent with the known error rates of the polymerases involved. Finally, you can ask about assembly. How well can you assemble a genome de novo? So everything I've done up till now has been based on mapping against a reference. You can say, let's throw out the reference. How well can you assemble a genome? And from the different amplifiers, in particular from these single cell measurements. And again, it turns out that the microfluidic MDA tends to be the best one, in large part because of the low number of contaminants in there. Contaminants really mess up the assembly process. And so after going through all this work, we decided to stick with what we started with, which was fortuitously a pretty well-performing amplifier. So let me now zoom back again and talk about some of the ways we're using this genome amplification to do single-cell genomics of humans. And you may ask, why bother? The average human genome is done. You can do a personal genome now. Each of us in the room can have our genome sequenced. Why would we want to do single-cell genomes? Well, you have to remind yourself that not every cell in your body has the same genome. And that's been known for decades, right? There's some really obvious examples. One being the immune system. You think about B cells, which are the cells that make antibodies. Each one, when they decide to make an antibody, makes such a strong commitment to that particular antibody that they reprogram their genome to hardwire that new gene in there, kind of like Cortez burning his boats when he got to the New World. There's no going back for those cells. They've really chosen to make that antibody. Another example are gametes, sperm and egg cells. They're all different because not only are they haploid, but they also, through the process of meiotic recombination, have created unique genomes in each individual cell, and there's a huge amount of diversity there. And finally, cancer, which you can think about as kind of the terminal end state of aging, okay, and cell division. Every time a cell divides, a few random mutations are accumulated, and so that creates genomic diversity. And if you happen to get the wrong set of mutations, the cells will proliferate without control and create tumors, which themselves have genomic diversity. And so that, of course, is interesting to explore. I'm going to talk about, I'm just going to talk about a few of these very briefly because much of this data is already published, and I'll just sort of point you to it. So the gamete cells, where we focused on sperm, allowed us to sort of explore this question about what is the recombination potential for any given individual. Now, this is a question that's been considered at length across the human species, and there have been very large projects led by HapMap and Decode and others to try to measure recombination diversity across human diversity. We decided to ask, what do you get when you look at that at this individual person level? In particular, how does those two measurements compare? This is the work of Jianbin Wang and Christina Fan in the lab. We published it in Cell 2 years ago. And we made sort of the very interesting discovery that, in fact, you get just as much diversity within one individual as you get pretty much across the entire species, which is kind of amazing. I sort of think of it as an ergodic theorem of recombination. And this touched off a lot of interest in this area, and so now people have gone and replicated our results. They've gone and looked at eggs, and it's creating a lot of interest in vitro fertilization. There's sort of very practical applications of this sort of thing, and it's sort of taken off in different directions. In the case of cancer, we got interested in what are the earliest mutations that lead to cancer and sort of the precancerous cells. And this was work we did in collaboration with Ravi Majeti. It was Tom Snyder from my lab and Max Jen from Ravi's lab. And we were looking at AML, acute myelogenous leukemia, as our sort of model system in humans. So it's the real naturally occurring human disease. And went through, and it turns out that there are, for these people who are very sick, it's a liquid tumor. Their, hematopoietic compartment has been trashed. There are a few residual normal hematopoietic stem cells, which by all the sort of phenotypic measurements appear to be normal, not part of the tumor. All the flow cytometry, surface markerology, and all that. We decided to look at that little subgroup and ask, do we see mutations from the tumor in that group of residual normal hematopoietic stem cells? And it turns out that we do. A very small fraction of them have mutations that are associated with the tumor. Those must have been the earliest mutations. And we went by doing single cell analyses. We were able to work out the exact order of occurrence of those mutations and show, more or less for the first time in a natural tumor, that there is sort of a unique, discrete set of mutations that happen. It's not parallel evolution. It was sort of one leading to another, and we were able to map that out in several patients. Ravi's gone on and done some really beautiful further work with that approach, and recently had a paper in PNAS which followed up on this and with really interesting results. And this is now something that in this case, we used a trick to do the single cell amplifications. We've now sort of ported it all to the C1 and are sort of deep into looking at other tumors and sort of the mutational spectrum at the single cell level and how different clones propagate. That's going to be, I think, a very fruitful field going forward. And finally, whenever I give this talk, somebody always raises their hand and says, all this single-cell genomic stuff is all well and good, but how about single-cell epigenetics and epigenomics? And my standard response has always been, well, that's a much harder thing to measure, which is a true statement. But we finally decided to take a whack at it. And through my collaborator, Bill Burkholder, and his group in Singapore, he and his postdoc developed a very nice single-cell methylation assay for looking at many genes in parallel, and using an old trick, methylation-sensitive restriction enzymes. They tuned that up to the single-cell level and sort of got a very beautiful result, which was published in Science last year. And so if you're interested in that, I'll refer you to it. And with that, I think I'll stop and summarize. Oops. This sort of emerging tidal wave of single-cell genomics is based on those three advances I talked about earlier on, amplifier technology, sequencing technology, and cell manipulation technology. And I hope, as I've gone through the talk, you've gotten a sense about how they all sort of interrelate and work together here, and how you can use them both to do phenotypic analysis, do transcriptome analysis, and genomic analysis in a wide variety of systems, ranging from eukaryotes to microbes. And I'll just stop now and take any questions you might have. Thank you.
Questions for Steve? Okay, I'll start with one. Almost about something you didn't talk about, but you spent a lot of time, you put a lot of effort into measuring single molecules and things in single cells. But one of your most impactful studies recently has been cell-free DNA sequencing, which is really, this is a way for taking maternal blood and doing genetic prenatal diagnosis. And it's, Illumina bought this company, and you went to Hawaii. And so,
with you.
Right. So how, it has nothing to do with single cells. It's
right.
very orthogonal thinking. And so how do you connect?
How did I get there? Well, how did I get there? I got there by becoming a dad, first of all, and realized this sort of very medieval thing about risking your baby's life to ask a genetic question was really not what you wanted to do. And I got there through just very simple ideas about counting molecules. And in my earlier life as a single molecule biophysicist, it was all about counting molecules. And so we'd spent a lot of time thinking about that. And as I realized, I should be thinking about things related to human health, and stumbled across this field of cell-free DNA, I realized they were hung up because they weren't counting molecules, and the sequencers turned out to be a good way to do that. And so that all came together. And that's led us down many other interesting avenues, not just in the world of prenatal diagnostics, but also in the microbiome, which we got pulled into that through our work developing a diagnostic for transplant rejection. And so we have now a non-invasive diagnostic for when heart and lung transplant patients are rejecting their transplant. And in doing a very large study to validate that, one of my postdocs, Ewan de Vlaming, realized that we had been sort of unintentionally sequencing a lot of microbiome data. 98% of the cell-free DNA in these people's bloods was human, 2% was from the microbiome and the virome, and he decided to focus on that. And we discovered a whole new world. And these were a very interesting cohort to look at because they'd all had their immune systems pharmacologically turned down. They were all taking immunosuppressant drugs, and we could ask what happens to the viruses and bacteria in their body as you turn off the immune system. The first chapter we published in Cell last November, and there's many more things coming out of that now. I mean, we're really having a lot of fun teasing all that apart. It's a very powerful data set.
Yeah, I think it is an example of very rapid, I think what Steve's been special at is being able to take laboratory insights or take clinical insights and convert them into really practical things that, scientists frequently, we add upon a pile of information that eventually becomes applied. Steve's been very fortunate that his work has very rapidly been applied. Jim?
I'm sure you hear questions about how single cells differ when you rip them out of a tissue and put them into a microfluidic device to measure their transcription. I'd like to turn that question around and ask whether you've started to rebuild tissues, putting sets of cells together, small combinations of cells together, and looking how those cell-cell interactions alter transcription.
Yeah, so this question of whether you can assemble tissues from different cell types is one that has received decades of attention. It's called tissue engineering. That's why I sort of tongue-in-cheek call it, we do reverse tissue engineering. And so a large number of groups around the world have been trying to do that with varying degrees of success. It's not something we've personally gotten into, but I think sort of the results of what we measure can be used to sort of more intelligently build up artificial tissues for sure. Yeah.
Okay, I have another question. You have these cells that look the same based on their external markers, and then you're able to show their different populations. But are there ways to use that information to find new markers or other way?
Oh, yeah. Yeah. So you can certainly make this virtuous circle where you take what you've learned from the transcriptome measurements and develop new markers based on that. There was one example that sort of went by very quickly. I didn't dwell on it in the colon stuff, but yeah, I mean, and it's been very, very useful in sort of tumors and cancer stem cell populations and things like that. And our friends in the lung are now going to be very busy doing that as well. Yeah.
Okay, well, I'd like to thank you, Steve.